of government contracts for Ology Bioservices, a contract manufacturing and development organization that specializes in products for infectious disease. Prior to his current position, he was president and chief scientific officer at Dyneport Vaccine Company, a biotechnology firm in Frederick, Maryland. He has also worked at Covance Labs in Madison, Wisconsin, and IIT Research Institute in Chicago, Illinois, where he managed highly successful programs in immunotoxicology research and testing. Dr. House has more than 20 years direct experience in U.S. government contracting. He earned his Master of Science in Public Health and PhD degrees in medical parasitology from the University of North Carolina at Chapel Hill School of Public Health. He is the author or co-author of more than 100 journal articles, book chapters, and books covering immunology, toxicology, infectious disease, and biodefense. He's an adjunct professor in the Department of Public and International Affairs at George Mason University and a fellow of the Academy of Toxicological Sciences. Sorry, choking on this. As I mentioned, he taught one of my final classes as part of my coursework for my PhD. He taught a medical countermeasures class. That was, as I said, one of my favorite. I still talk about it all the time. So I reached out to him. We were connected on LinkedIn, and I saw some of the work that he was posting about COVID. And I asked if he would be willing to speak to us. And he put together a great presentation on Tale of Three Pandemics. So I am going to pull it up, and then I will turn it over to you, Dr. House.
All right. And can I advance these, Jen, or do you do that?
I can do it. So just let me know whenever you want me to move ahead.
Will do. So thank you, Jen. Good evening, ladies and gentlemen. Since we are having a multiple admiration society going, I have to give props to Jen as being one of my favorite students, if not the favorite student. The reason is I always knew she would keep me on my toes. Nobody ever asked as many questions in my class ever as Jen. So at least I knew there was one person paying attention and enjoying it. When Jen asked me to do this presentation, I thought about it. What can I say that is not necessarily a dry recitation of facts, because 8 o'clock on a Wednesday night, you don't want to hear a whole lot of facts, although I'll give you a lot of facts. But rather, in the course of 20 years of doing medical countermeasure development, what have I seen that is an interesting trend? So to me, the idea that in the 8 or 9 years that I've been teaching medical countermeasure development, I kept saying, we're going to have another pandemic. It's just a matter of when. It's not if, it's when. And it's, students, I think, believed it, but now we are in a pandemic. And so I thought, so what can I draw that's interesting from that? And I thought, with apologies to Charles Dickens, maybe a tale of three pandemics. Three pandemics that really have been instrumental in history of the world, and how they may be different and how they may be alike, and what we can learn from that. So let me start out by saying, what is a pandemic? So a pandemic is, I'm having, let me see how I can need to pull away some people's pictures. There we go. So what is a pandemic? So as usual, a lot of people go to the dictionary. So a pandemic is an outbreak of a disease that occurs over a wide geographic area and affects a significant proportion of the population. We compare that with an epidemic, which is the beginning in a lot of cases of a pandemic. An epidemic being something that affects a disproportionately large number of individuals within a population, so it's more limited. It's easy to say then that pandemics almost always begin as epidemics, and they then expand. Now, pandemics have occurred throughout human history, and let me be careful in saying this, because you can say that, well, maybe pandemics occurred even in prehistory, but I think it's important that we recognize that pandemics really happen when we reach a critical mass. They are propagated by human travel and contact. And so in prehistory, we probably didn't have pandemics. They certainly epidemics, but not pandemics. So this is more of a, if you will, a more modern malady of humans. Pandemics evolve over time and they can become less severe or they can become worse. So tonight, what I'm going to do with your forbearance is to talk about 3 pandemics that I think are important for different reasons. Next slide, please. I want to put things into perspective to start with, because when you're in a pandemic, it's obviously the worst thing in the world. We've all been personally affected by COVID-19, but it's instructive to remember that this is not the worst that we've ever faced. Probably the worst, obviously, is plague. And plague, as you're probably aware, occurred in 3 major waves. The first was the plague of Justinian in the mid-500s. And this was significant because this is when plague really became a significant human issue. And possibly 30 to 50 million people died. Obviously, this far back, we can't tell for sure. We have to assume, we have to extrapolate. But 30 to 50 million people was half of the world's population. So that's definitely the first level of perspective. The one we're more familiar with, or most people are more familiar with, is the Black Death from the mid-1300s. And this was the granddaddy of all pandemics. This is somewhere between 75 to 200 million people, about a third of the European population. Now, what seems to get less recognition is the so-called third pandemic, which started in 1855 and could arguably be said to still be with us. And the third pandemic, which started in China, and probably most of these started in Asia, killed about 10 to 15 million people. So these are very sobering numbers just from one disease. The next pandemic that we'll talk about is the Spanish flu from 1918 to 1919, which killed approximately 50 million people worldwide. Again, really the second biggest killer of humanity after the Black Death. And then, of course, the pandemic that we're in now, COVID-19. And as of last week, at least, it had killed not quite 3 million people. So let's take these one by one. And I will warn you, ladies and gentlemen, I will be talking fairly quickly tonight because I'm notoriously bad at running long, and I do want to leave time if there's any questions. So next slide, please. Let's talk about plague, and this is the dance of death. You may have seen this woodcut before. Next slide. So plague is caused by infection with a bacterium known as Yersinia pestis. It's gram-negative, which doesn't matter, but it's intracellular, which does, because that means that this is a bacterium that's able to hide from antibodies by and large. It's also notorious because it's genetically plastic. That means that its genome can be modified to be antibiotic resistant fairly easily, whether by nature, by mutation and natural selection, or offensively by humans. As a matter of fact, this is an agent that has been weaponized by several countries. So it's not a theoretical threat. Plague still exists in reservoirs on all continents except Australia. Now, I see this everywhere I read it, and they never mention Antarctica, but I have to assume that it's not endemic there either. Please keep in mind, plague has never left us. It just laid low. Plague still exists endemically throughout the world. Next slide. Plague is a horrible, horrible disease, and it exists in 3 forms: bubonic, septicemic, and pneumonic. Bubonic is the most common in nature. Bubonic has the symptoms of malaise, tachycardia, abdominal pain, etc. And it causes a very characteristic sign, which is buboes, which are large enlargements of lymph nodes, generally in the groin and cervical area. And bubonic plague is 50 to 60% lethal if untreated. Now, that if untreated is very important because for most of these three forms of plague, if treatment is not initiated within 48 hours of infection, you're probably already at the point of no return. That becomes important in a few slides. The second form is septicemic plague, which is when the disease escapes the lymph nodes and gets into the bloodstream. And then the final terminal form is pneumonic, and this is the worst. This is where you have severe bronchopneumonia, you start coughing up blood, you have chest pain, shortness of breath. This is a rapidly fulminant disease. It is 100% fatal if not treated within 20 hours of onset. Plague generally has an incubation period of 2 to 6 days, and then you have the sudden onset of illness. But keep in mind, you're incubating it the whole time. Death from plague is due to systemic shock, disseminated intravascular coagulopathy, and organ failure. A very characteristic sign, as you'll see under septicemic form, is necrosis of the limbs. And this is probably why plague was called the Black Death, because you actually get necrosis and blackening of the extremities. Next slide. Now, treatment of plague, and you'll see this in any reference, is fairly straightforward with antibiotics. You can, if you expect to be exposed to plague, you can take antibiotics pre-exposure. Doxycycline works great. Tetracycline and others work almost as well. And for post-exposure, although the regimen of dosing is a bit more involved, it is still fairly straightforward. Gentamicin, streptomycin, chloramphenicol, tetracycline, and ciprofloxacin all work really well. Now, this is where we run into a divergence in thinking, because the civilian part of our government, HHS, more or less has deemed that in the event of an exposure to plague, we would treat everybody with antibiotics. My concern is, first, are there enough antibiotics to go around? And the second is, as I mentioned, the window of opportunity from exposure to treatment, successful treatment initiation, is really just a couple of days. And like most of the diseases that we're concerned about from a biodefense standpoint, and all three of the diseases we're going to talk about tonight, the initial signs are, quote, flu-like syndrome. So it could be anything. But again, plague can be treated with antibiotics very successfully. Next slide. Where we run into concerns, or at least my concerns, are vaccines. There have been vaccines developed for plague. The early vaccines were based on whole cell bacteria, whether killed or live whole cells. These early vaccines were safe, but they were effective against bubonic plague, but not pneumonic plague. As a matter of fact, soldiers in the Vietnam War received the plague vaccine, and they were protected against bubonic plague, which was endemic, and I think still is endemic in Vietnam. However, in the attempt to make this a better vaccine, certain entities developed a formalin-killed whole cell vaccine. Particularly, you're always concerned about live whole cells, and so the idea is that if you kill it, then it's much safer. Well, this was also proven to be effective against bubonic plague in Vietnam. However, this is a highly reactogenic vaccine, which means that it causes a lot of significant side effects. More to the point, the immunity provided by a killed whole cell vaccine was not very long lasting. I wish I could go into all of the gritty details that I regale my students with on why different vaccines are better or worse than others, but by and large, a killed vaccine is, in my mind, probably the least effective of the lot. Next slide. Now, moving away from the I guess, brute force approach of using killed organisms. USAMRIID in the United States and DSTL in the UK started working on a more rationally designed vaccine candidate. And whether through collaboration or competition, both parties came up with candidate vaccines. And I believe this was like in the 50s and 60s. These vaccines were both based on two factors. One is an antigen called F1, which is a capsular protein, and a low calcium response virulence factor, LCRV. Both of these vaccines were demonstrated to protect against bubonic and pneumonic plague in animals. That really should be underlined. They were different from each other in that the UK's vaccine put both of these vaccines, both of these antigens together. It was called F1 plus V. The United States developed a fusion vaccine that fused both of these together. Well, this was problematic because there are F1 forms of the bacterium or F1 negative forms of the bacterium that would not be protected against by this vaccine. LCRV antigen is not 100% protective regardless, and LCRV variants have been described. So the take home from this slide is that all of the chips were put into the basket of developing vaccines based on these two antigens. I will tell you that the F1V vaccine, I believe, is still under development, and it has been under development for more than 20 years. And that is perhaps accurate to say advanced development. Unfortunately, ladies and gentlemen, the denouement of all of this is we still do not have a credible candidate for plague vaccine. There are lots of other technologies being developed, but unfortunately, the US government is not funding a lot of research into plague vaccines anymore. So this is one of the first learnings, is one of the deadliest diseases that's ever affected humans. We have treatment as long as it's used quickly enough, but we don't have vaccines to protect people. So this is problematic. Next slide. So then the next pandemic we'll consider, to me, is the most important, or perhaps at least the most interesting, and this is pandemic influenza. Influenza, and I won't bore you with the details here, it is an RNA virus. There are 4 types, A, B, C, and D, with A and B causing human disease. Categorization is based on the nucleoprotein and matrix proteins, that is, the proteins within the interior of the vaccine that carries its genetic characteristics. Influenza A viruses, the ones that we generally are more concerned about for a seasonal standpoint, are further characterized by subtype, and this is based on 2 surface glycoproteins, that is, proteins that have sugar moieties on the outside. These 2 surface proteins are hemagglutinin, or HA, and there are 18 subtypes of those, and neuraminidase, or NA, where there are 11 subtypes. Of these, only H1, H2, and H3, and N1 and N2 have been associated with human epidemics. The HA consists of 2 subunits, HA1 and HA2. The important part of all this is that highly pathogenic strains of influenza allow activation of hemagglutinin in the absence of a protease. And what that means is we can use this as a target for reverse genetics in vaccine development. So the influenza virus itself has a key, which is hemagglutinin and neuraminidase. Next slide. The disease itself, perhaps not as well known as people would really think. I'm sure all of you have lots of incidents where people say, I was not feeling well over the weekend, I had the flu, but I'm better now. That's not the flu. Between 5 and 50%, which I think is extraordinary, of the US population contract influenza each year. Spread, as we know, is primarily through aerosol exposure. The primary target of influenza is the respiratory epithelium, although other tissues can be affected, but they don't make us as sick. Influenza is generally a self-limiting disease, about 1 week in healthy individuals. Here's the kicker. You don't get the flu over the weekend and feel better by Monday. Once you have influenza, weakness and fatigue can last for several additional weeks. So this is a very bad disease, even if you're a healthy individual. I will share with you that I have had influenza a grand total of 2 times in my life, the last one being a serologically confirmed influenza, which maybe apropos of nothing, I appear to have contracted in Las Vegas. It was the worst disease I can remember having. And it was also a turning point in my life in that I finally realized that, hey, maybe flu vaccines every year is a good idea. Like other respiratory infections, influenza is especially hazardous in those with pre-existing conditions like chronic pulmonary disease, immunosuppression, diabetes, pregnancy, and obesity. You'll notice all of these conditions are those where the immune system is compromised. That becomes very important in a moment. Next slide. Now, flu can have severe complications even in people who are not at risk. These include hemorrhagic bronchitis, diffuse alveolar damage, and pneumonia. Generally, the pneumonias associated with influenza are not viral. They are generally caused by bacterial infections, and most deaths and significantly serious cases of influenza are associated not from the virus itself, but from the bacterial pneumonias. Can you go back a slide, please? Thank you. People 65 and older, although making up about 15% of the total US population, account for more than 90% of deaths in seasonal influenza. Now, I've just told you that seasonal flu kills you from pneumonia. Unlike seasonal influenza, pandemic influenza is associated with induction of something called a cytokine storm. And that basically is putting the immune system in high gear with essentially no breaks associated. And this runaway immune response causes a significantly enhanced inflammatory response, which is generally where pandemic flu exerts its worst effects. Next slide. So how do we go from seasonal to pandemic influenza? 2 topics for you to be aware of. One is antigenic drift and antigenic shift. In the interest of time, I won't go through the entire thing here, but it's important to know that the influenza virus has a polymerase that is an enzyme that allows it to replicate its own RNA. And this polymerase lacks the ability to proofread. Because of this, the virus is very susceptible to frequent mutations, which it is unable to repair. Antigenic drift results from minor changes in the HA and NA proteins that I talked about. And this occurs pretty much all the time. Flu picks up between 3 and 4 amino acid mutations every year. And so every 2 to 5 years, you get a major drift due simply through an accumulation of mutations. At this point, pre-existing immunity is generally incompletely effective against new strains. Antigenic drift, put shortly, is why we have to get a different influenza vaccine every year. However, once in a while, we see what's called an antigenic shift. And what happens then is this results from new hemagglutinin or neuraminidase subtypes that emerge in human populations without pre-existing immunity. This occurs when the various viruses recombine or reassort within an animal reservoir. This generally occurs within birds and within swine. Next slide, please. There have been numerous pandemics associated with influenza. In the past 500 years, we believe there have been about 14 influenza pandemics. But I want to just bring your attention to 4 because I think they're the most interesting. Spanish flu of 1918, I'll talk about that much more in a few minutes, but the important part here is that this was caused by H1N1, and this was associated, as said previously, with 40 to 50 million deaths worldwide within a period of about a year and a half. There was an Asian flu of 1957 that had about 1 to 2 million deaths. Hong Kong flu of 1968, about 500,000 to 1 million deaths. And swine flu, the most recent one of 2009, believed to have caused about 284,000 deaths. Now, if you're somebody who likes to draw graphs, you're looking at this and thinking, well, the 4 most recent influenza pandemics seem to have been subsiding. So maybe pandemic influenza is not that big of a deal. But that's not the case. Next slide. So let's talk for a minute about the 1918 flu and why that was such a big deal. It was called the Spanish flu, but the first outbreak was actually in Fort Riley, Kansas, and it appears that the global pandemic appears to have started in France. You can read up if you want to see the stupid reason it was called Spanish flu. It really was kind of dumb. This, we believe, resulted from the direct transmission of the H1N1 virus from birds into humans. Any time that you get transmission of an influenza virus from birds into humans, and I won't go into the biology behind why this is, but once that happens and you get sustained transmission between humans, that will almost always result in a pandemic. Spanish flu occurred in 3 waves between 1918 and 1919. The first was relatively minor. However, the second wave, which was the worst of the 3, was widespread illness and high mortality. And then the disease sort of petered out in the spring of 1919. It was widespread, but by this point, we'd started developing herd immunity and there were much fewer deaths. Please note, only the Black Death was known to have killed more people in a similar span of time. Spanish flu killed almost 700,000 people in the United States, and this was more than the troop deaths in World War I and World War II combined. So you can see Spanish flu was really the worst pandemic that we have experienced, and hopefully the worst we will experience. Now, this virus, this H1N1, was not particularly infectious or lethal. What made this unusual is that the target population was males 18 to 40 years old, and they were differentially affected. This was resulting in a so-called W-shaped mortality curve. It was atypical, therefore, in that those who had the strongest immune system were apparently at the greatest risk of death. We know now, or we think we know, that the immune response to the infection was the cause of the great mortality of Spanish flu and not the virus itself. The pathology was characterized by lung inflammation, and many of these deaths appear to have been due to secondary bacterial infections following this dysregulated immune function, the cytokine storm, which caused inflammation, which led to secondary bacterial infection. Next slide. So what is the next pandemic going to look like? Well, we like to think that we can't predict them, but we believe that it's likely to be the H5N1 variant. You'll notice in parentheses, this is an avian virus. We already know from human exposure that this is highly lethal. For people who are infected with H5N1, the case fatality rate is about 60%. Luckily, this virus has not yet exhibited effective person-to-person transmission. We do know that several amino acid changes that were in the Spanish flu are present in H5N1. So it carries the, if you will, it carries the germs of the next pandemic based on the worst pandemic. The changes that we're seeing are consistent with those that are associated with human adaptation of this virus. This virus also appears to exert pathogenicity by inducing a cytokine storm. And influenza is still endemic in waterfowl in China and will always continue mutating. Human exposure appears to be associated with direct exposure to poultry and other waterfowl, and environmental exposure, including swimming, bathing, in virally contaminated water. One of the things I like to say in my course is that we are literally swimming in a sea of viruses every day. With influenza, this is literally true. Next slide. There are 4 currently licensed drugs for influenza. You see the 2 at the top are in red. These were 2 that have already, the circulating viruses have already developed resistance to, and these are no longer used. So we have Tamiflu, Relenza, Rapivab, and Xofluza. And even as recently as a year ago, I heard people say, well, doctor told me to get Tamiflu. Well, they probably won't this year because now we know that Tamiflu and Relenza are both now being shown to have viral strains becoming resistant to them. My point here is that we will always have to continue developing new flu drugs. It's not like antibiotics that will work pretty much for decades. Flu virus drugs will always require new drugs on the horizon. Next slide. There are a few other drugs under development. The one that I will point you to that's most interesting to me is Fludase, being developed by Ansun Biopharma. This is different. Instead of inducing antibodies against the HA and NA like vaccines do, or interfering with hemagglutinin synthesis like the drugs in the previous slide, this drug is a recombinant sialidase. And what that does is it removes sialic acid from the surface of lung cells. That is the proximate receptor that flu virus binds to, and that's how it gains entry into lung cells. This is a rare example of host-directed rather than pathogen-directed medical countermeasure. And I'll talk a little bit more about that in my last slide. Next slide, please. In terms of vaccines, there are 2 types of seasonal flu vaccines in use in the United States. These are trivalent inactivated vaccines and quadrivalent inactivated vaccines. The basis of protection is primarily demonstration that you're inducing neutralizing antibodies that neutralize the hemagglutinin. You can do other types of assays, but this is considered to be the gold standard. Now, the reason this is important, if you develop, for example, a measles vaccine, if you develop a second measles vaccine, it's going to take you almost as long to develop that second measles vaccine as with the first. The question is, why doesn't it take 10 years, the generally accepted time frame for developing a vaccine? Why does it not take 10 years every year? Why are you able to do this yearly? And it's because we know that a titer of 1 to 40 for the hemagglutinin neutralizing assay has been shown statistically to protect people at risk of influenza. So this is one of the benefits of really understanding what your target is, hemagglutinin. But hemagglutinin also has some tricks up its sleeve that I'll talk about shortly. Next slide, please. Because we have to get seasonal influenza vaccines, and because we are always going to be under the threat of pandemic flu, why can't we just develop one vaccine that will protect us against everything? Well, we're trying. This is called a universal influenza vaccine. Current flu vaccines contain either 3 or 4 subtypes. And as you probably know, these are selected by WHO based on global surveillance, which means that we have to predict every year in advance, not just for one hemisphere of the world, but for both, North and South. And sometimes these predictions have been wrong. This also requires development of modified vaccines each season, which is very time, labor, and resource intensive, not to mention the fact that compliance tends to be problematic. So we need a way to break this cycle, and most importantly, to develop a rapid response to novel pandemic strains. Next slide. A universal flu vaccine, according to WHO, should be at least 75% effective, should protect against both group 1 and group 2, which are the types that we're most susceptible to, should have a long duration of protection, at least a year, and it should be suitable for all age groups. Now, if you look at the insert sheets for all of the seasonal vaccines, they're all over the place. Some for children, some for elderly, some for 2 to 49, it's all over the place. So a universal flu vaccine would tend to make it much more stabilized. How do we do this? Current vaccines, as I've said, are designed to induce a strong immune response to the hemagglutinin molecule. Universal vaccines would utilize immunogens that are much more highly conserved. I told you that hemagglutinin comes in lots of different subtypes, and they recombine every year, and that's why we have to mix and match. A universal flu vaccine would find the portion of the virus that does not change, that is not susceptible to mutations. Some of these include the hemagglutinin stalk, the extracellular domain of M2, which is a portion of the virus that doesn't mutate, and neuraminidase. All of these, unfortunately, are relatively weak immunogens. So this has been one of the reasons that development of a universal flu vaccine has taken so long. Next slide. Some of the others, and I won't belabor this, are the ectodomain of the M2 protein and something called the HA2 protein. Also, the internal antigens, the nucleoprotein and the M1 matrix that I told you earlier are the proteins that basically bind up the RNA inside the virus, are also potential targets. So we're not suffering from targets. It's just these are generally protected in a natural infection from the immune system. So we've got to figure out tricks to make the immune system see these conserved antigens. Next slide. There are a number of promising candidates. Recombinant vaccines, which I'm generally not a big fan of, but these appear to be developing well. FP011 by Altimmune. This has 6 synthetic peptides that are linked to an inert fluorocarbon chain. And this is aimed at generating T cell responses to a wide variety of influenza strains. One that appears to be most promising is M001, being developed by BeyondVax. And this is also a recombinant protein that has 9 conserved epitopes. And this likewise is aimed at inducing T cell responses as well as enhanced B cell responses to a variety of different flu strains. Next. There are vaccines based on the influenza virus itself, one called Codavax, which is a live attenuated vaccine. Now, I won't go into this very long explanation of this using an algorithm to de-optimize the influenza HA and NA gene segments. Basically, what you need to know about this is a vaccine that was designed from the molecular level out instead of how we generally do vaccines where we identify protective antigens and then try to figure out after that what the sequence is. Like the others, this is aimed at stimulating an immune response against flu for multiple seasons and, in this case, multiple decades. And then Ready Flu, being developed by FluGen, This is a novel single replication platform. This is to protect against both influenza A and influenza B viruses. This also is different in that it's administered intranasally and it's aimed to elicit cross-reactive antibodies. Most of the others are aimed more at cellular response. Ready flu, I think, is interesting because this actually is trying to crack the nut of antibodies not being very effective against these conserved epitopes. Next slide. Finally, nanoparticles and nucleic acids are being developed. This last one I think is very interesting. It's a microconsensus DNA vaccine. And this is very similar to the other vaccine that I was talking about that takes a different approach of starting at the molecular and sequence level and building a vaccine from the inside out. This is likewise delivered, this is delivered intradermally or intramuscularly by electroporation, which is the downfall, I think, of DNA vaccines. I think eventually we will begin seeing RNA vaccines as influenza candidates as well. Next slide. So in the home stretch, the current vaccine, COVID-19, this will be a shorter portion of my talk because there's not as much known about this. But I will tell you that we were warned. COVID should not have surprised us. And there are a number of people, Ralph Barrick being the one that comes to mind most to me, who, when SARS first came on the scene, they recognized that coronaviruses had a lot of potential to really do some damage in the future. But we didn't pay attention to SARS, and SARS kind of went away by itself. Then we had MERS, the Middle East respiratory syndrome virus. So MERS came along to tell us that we're still here and we're still capable of causing disease. As a matter of fact, MERS was much worse disease than SARS, but like SARS, it went away on its own. So now we have COVID-19 or SARS-CoV-2. This, ladies and gentlemen, is now getting into the theme that I wanted to sort of wrap up in a few slides my talk tonight, is that for all of recorded history, we've known that pandemics are going to come around. How can we not be surprised again? So next slide, talking about COVID-19, positive sense, single-stranded linear RNA virus. Lots of information here that you can read later. Most important to me, this appears to be a zoonotic disease. The reservoir we think is bats. We all know that it was first observed in Wuhan, and human-to-human transmission is via inhalation of droplets and surface contamination. If you don't remember anything else about the biology, know that it has 4 structural proteins: the spike, the envelope, the membrane, and the nucleocapsid. And the spike protein is the one that we're interested in. This is the protein responsible for the virus being able to bind to host cells via a molecule known as ACE2. And this is the target for medical countermeasure development, including vaccines and antibodies. Next slide. This disease is generally asymptomatic following infection. Once it begins to be established, the infamous flu-like symptoms. I think it's funny tonight, the only disease that really deserved to have flu-like symptoms was actually flu. Following this initial stage of the disease, it progresses to acute respiratory distress syndrome, or ARDS. This is where you start getting diffuse alveolar damage, which is really the hallmark of the coronaviruses. As this disease progresses, the virally infected pneumocytes, the lung cells, begin releasing a variety of cytokines and pro-inflammatory molecules. This is the so-called cytokine storm that I've talked about previously. This attracts neutrophils and T cells. which are recruited by the immune system to try to dampen down the viral infection. And unfortunately, this is counterproductive because this massive influx of inflammatory cells results in a pronounced inflammation, which results in further tissue damage. This is the pernicious thing about COVID disease is the body's working to rid itself of the virus, and in severe cases, it ends up doing more damage than it does good. Next slide. So in terms of vaccines, we all know the vaccines that are coming out now. Nucleic acid vaccines. Pfizer and Moderna have the messenger RNA. Inovio has their candidate NO4800, which is a DNA plasmid, and others, including Arcturus, are developing other versions of this. I will tell you parenthetically, in the years that I've been teaching my class at George Mason, I always talked about nucleic acids as being a vaccine platform that I thought had a lot of great promise, but there were so many detriments to it that I thought it would be a long time. And so I will share with all of you tonight, I am humbled by the fact of my own stupidity in never being able to recognize that nucleic acids would in fact be the way that we start breaking the stranglehold of COVID-19. Next slide. Now, a lot of people don't want to get to 2 shots of a vaccine. And as Jen and I were discussing before everybody joined, plenty of people seem to think that nucleic acid vaccines are going to reprogram your genome, or they're going to insert microchips or whatever. So interestingly, more people seem to be really anxious about getting viral vector vaccines, which I find really ironic because nucleic acid vaccines, the reason you have to take 2 is that they go away on their own. The body is able to degrade the RNA. Viral vectored is a vaccine that is able to replicate. So the lead candidate, of course, being developed by AstraZeneca and Johnson & Johnson. These are both based on adenoviruses. Vaxart is developing a viral vectored vaccine a little bit further along, but it has the advantage of being orally delivered, a vaccine you can take by pill. There are a number of companies or entities working on recombinant vaccines. These, I believe, are all much further down the pipeline. They probably will have some role to play in future response to coronaviruses. And if the scientists are right and SARS-CoV-2 is something that we're going to have to live with from now on, a number of these vaccines will be available, just as there are now 10 or 12 seasonal influenza vaccines. But for my money, the nucleic acid and the viral vector will be what really go the long way in helping us get this under control. Next slide. Some therapeutics have been developed, remdesivir, various anti-parasitic disease drugs, glucocorticoids, and what I think is interesting, synthetic human vasoactive intestinal peptide, which is another one of those host-directed therapies that helps break the inflammatory response. By the way, you won't see some of the drugs touted by the previous administration and others that turned out not to be what they were sold as. Next slide. There are also a number of monoclonal antibodies being developed. There are some that are neutralizing, like bamlanivimab, casirivimab, and imdevimab. Both of these are under EUA, emergency use authorization. Vir is developing a neutralizing antibody that is dissimilar to the others. And then you've heard me tout this several times, host-directed therapies. There are 2 monoclonal antibodies that I won't embarrass myself trying to pronounce, but both of these are important in that they are against not the virus, but against granulocyte macrophage colony stimulating factor, one of the key cytokines in the cytokine storm. So I'm very happy to see a sort of a 1-2 punch, monoclonal antibodies directed not just against the virus itself, but against the inflammatory response as well. Next slide. So that was my rapid run through, or my rapid response, if you will, of a very brief comparison between what I think are three important pandemics. But the question is, and the question I want to pose tonight is, are we ready for the next pandemic? Because echoing my standard line from my course, there will be another. It's not a matter of if, it's a matter of when. And I think a lot more people understand that now. So next slide, let's talk about this. For plague, what did we learn? Well, medical biology was not advanced enough for us to really learn much back then. There were no effective interventions available. Dr. Schnabel did absolutely no good. But we do know, and we knew then, that increased mobility spread the disease, and that socioeconomic conditions favored virulence of the disease. Paradoxically, and I find this fascinating, the mass deaths associated with the Black Plague changed living conditions that favored the disease. And so it was actually the deaths themselves, not the fact that they killed people, but that it changed socioeconomic and social conditions so that people were no longer crammed up under each other, giving the bacterium easy passage between individuals. But what did we forget? Well, we forgot about plague. It didn't forget us. Plague is still with us. And I think, in my opinion, we're very short-sighted in depending entirely on antibiotics. I personally believe that we do not have any good vaccine candidates, but I also personally believe that we really need to have a vaccine against one of the deadliest diseases in human history. Now, although future pandemics seem to be unlikely, plague is still, I think, an ideal biological weapon. So we need to pay attention to this one. Next slide. For influenza, what did we learn? Although medicine was still not up to the challenge of developing antivirals, it was due to diligent public health efforts that the pandemic was able to be contained. We also learned that the avian N1 strain was going to get us regardless of what we did. We know now that H1N1 and any strains that originate in birds are going to infect humans. And once we learn how to spread it, then we're going to have another pandemic. So what did we forget? We forgot that some conditions favor viral recombination. The way that poultry are kept in certain parts of the world in close connection with humans still favors this recombination that results in antigenic shift. We learned that proactivity beats reactivity. We've now learned that having vaccines beats not having vaccines. And more importantly, we learned that antigenic shift means that there will always be influenza pandemics. Unfortunately, we've acted like that wasn't true. Even the 2009 pandemic, even though a number of people were waving the flag, we still had a very protracted and I think ineffective response to that. And next slide. So for COVID-19, what have we learned? What have we relearned? I think this is the slide I want you to take home with you tonight. Science is powerful mostly when it's unleashed. We went from a dogma that it takes 10 years to develop a vaccine to developing multiple vaccines in less than a year. So science unleashed can do great things. I believe that technologies currently under development are going to radically shorten this even more. I believe, and this just may be my personal belief, the definition of altruism, unfortunately, is starting to morph. I believe that we as a society do not take the long view for human health. I'm not going to preach about masks. I'm not going to preach about social distancing, but I think that what I've seen is people are more interested in protecting themselves than in protecting society at large. You're free to argue, but that's my personal opinion. I also believe that science by itself is inadequate without social engineering. As a corollary of that, what did we forget? We forgot that not everybody is a scientist. A lot of people don't want to be confused by the facts, which is harsh. I will share with you that sometime last year, Anthony Fauci made some comment. I don't remember what it was, but I remember being with several colleagues and commenting that I thought that what he said was tone deaf. It had something to do with how people, oh, I know what it was. It was people should just do what they're told. And that struck me as very tone deaf. We need to educate. We need to communicate. Not everybody's a scientist, and Lord knows I'm happy that they're not. But we cannot disdain people who don't understand science by saying, you just don't understand, you're stupid. We need to explain how an RNA vaccine works, and we need to explain how a viral vector vaccine works and why one is better than the other. We've also learned, but we forgot, that collaboration between scientists beats competition every time. We've seen extraordinary collaboration, but I fear that once COVID-19 passes as a pandemic, we'll go back to competition. The final thing is good, fast, and cheap, known in program management as the triple constraint. When I worked at Covance, that was our internal joke. You're developing a drug, you're developing a vaccine. You can develop it good, you can develop it fast, you can develop it cheap, but you can't do all three. You can get to pick two. I will tell you that the US government does not believe in this, but ladies and gentlemen, in 20 years of doing this work, I have found it to be absolutely the case. So Jen, I believe that may be my last slide. No, this is my last slide. So new tools in the toolbox. And we've talked about this throughout this course. Genetic vaccines have gone from being the redheaded stepchild to being the vaccines that are going to break, I think, this pandemic. Viral vector vaccines, a couple of years ago, very much newcomers. Now we see vaccines based on vesicular stomatitis virus, adenovirus, measles virus even. Monoclonal antibodies, certainly coming to the fore in controlling infectious diseases. There's even a concept called DNA-launched antibodies, where instead of injecting the messenger RNA or the plasmid DNA for the antigen, we just figure out what the sequence is for the antibody. We encode that and inject that, and instead of having to make expensive monoclonals, the body does that for you. In other words, you cut out the entire middleman, not only of the drug industry, but the immune system itself. And finally, one that I've talked about tonight that I'm really bullish on are these host-directed therapies. The bugs are always going to try to get a head start on us, so we need to have other tools in our armamentarium. And I think a combination of drugs designed to affect not just the pathogen, but to help the immune system along are going to be how we beat some of these future pandemics. So that's my last slide. No idea, Jen, if I went long, if I went really long, but if people are interested, I'd be happy to entertain any questions.
Well, I actually have a number of them that have come through. Let's see. When you say that pandemics arrive when population reaches a critical mass, what defines a critical mass? Global populations keep growing despite previous pandemics.
Okay, fair point. So if you think about Neolithic period, prehistory, My point was that pandemics likely didn't exist in prehistory because we didn't really have a global population. We had isolated groups of individuals. So in prehistory, obviously epidemics would be possible. But to me, based on the strict Webster definition of a pandemic, it really becomes a much more widespread disease that is more geographically spread out. And so again, in prehistory, humans existed, but it's not like we had a global population. It was a bunch of individuals. So I may have confused it more, but that's, I think certainly now with our global population, epidemics always are going to have the possibility of blossoming into pandemics.
Okay, although we hear about fewer cases of the plague and treatment of the plague is possible through antibiotics, are you aware of any resistance to any of the antibiotics currently used to treat it?
I am not aware of any that are problematic. My point in that particular slide is that due to the genetic malleability of Yersinia pestis, they make an easy target for people to nefariously engineer antibiotic resistant forms. So that's one of the reasons that I think at least having a vaccine available, or at least a vaccine candidate available, if nothing else, as an adjunct to antibiotics, gives us a leg up. But to answer the question, no, I'm not aware of any that have been problematic. They have been described, but not anything that's problematic.
Here's a good one for you. Why are you not a fan of recombinant vaccines?
I'm not a fan of recombinant vaccines for a couple of reasons. One is they generally take a number of administrations. In my mind, there are very few recombinant vaccines that are single shot. They almost invariably require adjuvants, and I'm not a big fan of adjuvants. You know, what I think are ideal vaccines are vaccines that mimic as closely as possible a natural infection. So a virally vectored vaccine to me is a really good vaccine because it tricks the body into thinking that it's been infected with the pathogen that you're interested in. Uh, Live attenuated vaccines, although they're problematic because of potential reversion to virulence, to me, those are still better than recombinants. Now, recombinants certainly have a lot of things going for them, and there's a lot of really successful and effective recombinants. I like recombinants better than killed vaccines, but if I were to rank them, a virally vectored vaccine to me is probably top of the heap, followed by live attenuated if you can prove that it will never revert. That's a long answer, but hopefully it's complete.
So why can the Vaxart vaccine be delivered orally but not the others?
It's encapsulated in some way, and I don't really know enough about the Vaxart vaccine to give a deep answer on that. Sorry about that.
Okay, so if some influenza deaths occur due to an overactive immune response, how do vaccines, which strengthen the immune response, work with those?
Can you repeat that one? That's tricky.
If some influenza deaths occurred due to an overactive immune response, how do vaccines, which strengthen immune response, work with those?
Okay. So I think what was happening with and it was the pandemic specifically, the pandemic flu, the Spanish flu, where we most associate the cytokine storm. And there was something about that particular virus, and I'm blanking on what it was, but there was something about that virus that allowed it to activate the immune response. a vaccine against a influenza virus is going to be directed generally toward the antigens and not toward anything that would activate the immune system. So for example, the hemagglutinin or even some of the other, I'm blanking here, the universal flu vaccines, they're all aimed at antigenic constructs that would not be responsible for activating the immune system. So those vaccines would counteract the virus before it had a chance to activate the immune system. That's kind of convoluted answer, but pretty sure that's accurate.
Okay, so those were the most of the questions. JD, did I get all the ones that you had? I know you had put a couple in there. Did I get the ones that you had asked?
You got the two most important ones, so that's good. Thanks.
Do you want the other? Did I?
They're fine. We're good.
Okay. What conditions will it take for current COVID vaccines to lose their EUA status?
I think they'll have to go through additional long-term safety testing, even after licensure, by the way. And I think the more people that get immunized and we start getting what are called field results. So up to this point, the EUA was based on highly controlled clinical trials. And I think to gain FDA licensure, I think there'll be three things that be necessary. Continued safety testing, actual field data, that is epidemiological studies at large. And I think eventually to achieve licensure, I don't know this for a fact, I don't speak for the FDA, but I think at some point we're going to have to determine what the correlate of immune protection is going to be. Since it's unlikely we're going to be doing human challenge studies with this virus, we'll have to have some surrogate. You recall when I was talking about the seasonal influenza, a hemagglutinin titer of greater than 1 to 40 is now established as the correlate of immune protection. So we'll need to understand what that is, and I think we'll need to understand better how these vaccines actually protect people. We think we know how they work. We know how they're supposed to work, but there's a lot more data that has to be gained. Now, that being said, it would not surprise me that most of these vaccines gain licensure. I'm going to go out on a limb and say by the end of this year.
Sounds good. So do you believe the general public will be open to vaccines that utilize nanoparticles, even though there may be hesitation towards the liver and spleen to effectively eliminate the nanoparticles?
Now that's an interesting question from a couple of different standpoints. I think people who understand science to a deep level may have some concern, right? Because and this is near to my heart because a couple of years ago I wrote an entire book chapter on immunotoxicity of nanotechnology, and so there is that concern with nanoparticles, with nanotechnology, that you start filling up the reticuloendothelial system. So I think people who understand it may have a concern about it, but I would also hope that people who understand that also would be interested in looking at the toxicological data, because that type of product would get some pretty significant scrutiny by the FDA for that very reason. Now, it's interesting from a second point, and Jen and I were talking about this again before everybody got on the line tonight. And Jen, if you don't mind me repeating your question to me, is
go ahead.
Given the concern that so many people seem to have that RNA vaccines are going to reprogram their DNA, how much harder is it going to be to convince them to take a DNA vaccine? So If you don't know what RNA is, at least you've probably heard of DNA and have an idea of what it is. But again, not everybody's a scientist, so not everybody knows what RNA is and how it relates to DNA and blah, blah, blah. So I think back to the question at hand, there may be that reluctance. If you talk about the public, I would not see the public in quotation marks as being tremendously concerned about nanoparticles, unless there was some just like unexpected toxicity that was captured by the news media. These are great questions, by the way. Enjoying this.
We are getting a lot of good ones tonight. Does anybody else have any questions? And actually, if you don't want to type them, you can just talk. Normally I watch the chat as the presentation's going on, but since I was screen sharing, I couldn't do it this time. So they just kind of rolled in and then I read them all. But if anyone has another question, you can just unmute and ask.
Go for it.
Yeah, I have a question about the plague. So how are we tracking those events? Are they being reported to departments of health, or how do we know those cases are still going on out there?
So a couple of ways. Plague is problematic from an endemic standpoint in places like India,
Okay.
China, few other places like that. And so the World Health Organization and other international health organizations track those. They remain endemic and likely will continue to do so. Now, in the United States, it's a little bit different because plague is endemic here, but generally only in the southwestern part of the United States.
Mm-hmm.
And we already know this. I apologize for repeating it. I think it's
That's
interesting. So plague exists in the southwestern United States, and the reservoir is prairie dogs.
Okay.
Plague does still pop up, and it's actually dozens or even scores of cases every year that plague shows up. And generally happens when people are out walking their pets. Pets get bitten by a flea that recently was on a prairie dog that had plague. What makes that a little bit concerning is that if you're a physician in that area, you kind of know to look for plague, but new physicians, and this is a broad generalization, but new physicians just don't get taught about plague anymore and what to look for. You know, people in Arizona and that area, that they recognize plague, but it is still tracked, I think, through Morbidity and Mortality Weekly. I think it still appears there in the United States. So yeah, we keep an eye on it from an epidemiological standpoint. We know it's still here, but it's unlikely to become widespread simply because of the reservoir.
Okay.
Not saying it couldn't jump into other animals. It's just kind of a weird case where the prairie dogs decided that plague was a good thing to have.
Interesting. Thank you.
So are universal vaccines really possible?
If you had asked the question, if you'd asked me 5 years ago, I would have had a quick answer, which was doubtful. And I don't know if you know Mike Carilla. Dr. Carilla is a MD, PhD, graduate of Duke University, although I never really held that against him. And he was head of division of infectious disease at NIAID. And by way of that, Mike Carilla was one of the smartest people that I've known in my career. And he told me categorically he did not think it would ever be possible. So 5 years ago, I would have just been his parrot. I think now technically it will be possible. And by that, I mean, I think we're discovering some really good potential candidates, the headless stalks for neuraminidase and hemagglutinin and some of these others. They'll be different than the existing vaccines because since most of these are not going to be aimed to inducing antibodies, but rather T cell responses, I think they'll look different from the vaccines that we have now. And what makes them problematic, and I think why Dr. Kurilla believed that they weren't possible, is it's not so much the technology, but it's the infrastructure. What assays do you do to prove that it works? How can you have a vaccine that you can continually test to make sure that it doesn't change if you don't fully understand how it works? So my long answer, even longer, yes, technically, scientifically, I think they are completely possible. I don't think they're on the imminent horizon. My guess is at least another 5 years before we see vaccines that are close to universal. I think much longer than that before we see one that is one shot and done and you never get flu again.
Thanks.
Very interesting.
Okay, do we have any other questions? I know we're running long. We've got, as long as you don't mind. Any last questions? It doesn't look like it. So thank you again for coming and speaking with us tonight. This was a great presentation. I'm getting some feedback in the chat. Everyone's saying excellent presentation and thank you so much for your time. So we really appreciate you coming and talking tonight.
The pleasure has been mine, and everybody, I appreciate you sticking with me this long. Jen, thank you for the invitation. It has been one of the high points of my career to see a student achieve what you have. So tonight has been very special for me as well. Thank you.
Thank you. Well, doesn't look like there are any last minute questions. So just last reminder that last webinar of the spring semester is going to be May 5th at 8 o'clock. And if there are no last minute questions, then good night, everybody. And thank you so much for coming.
Thank you.
Bye.